So we will continue our discussion with recombination. And in this segment, as I mentioned earlier, we will discuss about few classical microbial genetics experiments where using phage, people are able to identify different aspects of recombination. So I'll share my screen now. Okay. So the evidence that recombination experiments can be conducted by bacteriophage came with several observations. One of the major observation came from Matthew Meselson and Jean Wegel. So what they did, they they actually took two different phage, one phage with two specific markers, presumably two specific genes of one kind of allele, and another phage with two specific genes with another version of the allele. What they did for the first phage, they isolated the phage from a bacteria which was grown on media with C12 and N15 isotope of carbon and nitrogen, which means that the DNA, I mean the phage DNA, that would be there on plate, those will be labeled with the light isotopes. The second phage they isolated were from a media where the carbon is C13 and the nitrogen is N15. Both are heavier isotope of carbon and nitrogen respectively. So they grew these two phage in two different media. They isolated these two phage from two different media, and then they co-infected a host E. coli cell with these two phages simultaneously. So the genomic DNA from both the phage will get into the E. coli cell, and if they recombine, then there will be some DNA which would be labeled both by light and heavy DNA. But if they don't recombine, then the DNAs will be either heavily labeled or it will be lightly labeled by the isotopes. The principle that they adopted to analyze this was similar to the classical Michelson-Stahl experiment, which was exploited to identify the semi-conservative nature of replication. So there they used cesium chloride density gradient centrifugation to separate differentially labeled DNAs. Here they used similar kind of density gradient centrifugation. So if you perform some density gradient centrifugation with three versions of DNA, one lightly labeled, one heavily labeled, and one mixed, what kind of band would you expect? One will equilibrate, the heavily labeled will equilibrate with denser part of the gradient. The lightly labeled will equilibrate the lightest part of the gradient, and then the mixed one would be in the intermediate phase, in the intermediate kind of distance in the density gradient. So what they observed is after this experiment with these two phage, they found out that indeed there are intermediate bands which indicates that some kind of a recombination event would be there, because the intermediate band means that the DNAs were exchanged, and that's why some part of the DNA may be lightly labeled and some part of the DNA may be heavily labeled. So this was a kind of early observation, and when the markers were analyzed, it was found out that indeed there is a mixing of the genetic markers, like we have already observed similar kind of crosses for meiotic recombination, and here something like the marker exchange is occurring between the DNAs. Now, second observation came from an experiment which is called the Hershey's cross. Here, the system that was used is a host E. coli And the phage that was used is T2 phage. Now for T-even phages, a specific type of phenotype is observed. This phenotype is based on the typical characteristics of the plaque they produce on bacterial lawns. What are plaques? Plaques are clear zones on bacterial lawn which are produced by lysis of the bacteria by some virulent or temperate phage. So this T2 phage, it would infect a bacteria and then it will lyse. However, it was found out that there are mutants of this T2 phage which shows variation in the plaque characteristics. So some plaques, the typical plaques of the T2, they are kind of cloudy and they are a bit smaller. But for the mutants, they found out that some plaques can be cloudy and larger, some plaques can be clear and larger, and some plaques can be clear and small. So they assigned these two characteristics of cloudy nature and the size of plaque as H for the hazy or cloudy nature and R for the rapid lysis nature. So rapid lysis means if R is plus, it would be a smaller plaque. If R is minus, the plaque will be a larger plaque. And if H is plus, the plaque will be a cloudier one. And if H is minus, the plaque would be a clearer one. Okay. So if this is the situation, and if as per the experimental model, if you infect the bacteria with a phage with H minus R plus characteristics, which is H minus R plus means which can produce clear and small plaques, with another phage with H plus R minus characteristics. So H plus R minus is cloudy and large plaques. And if you obtain some of the plaques like cloudy and small, and some plaques clear or large like these two H plus R plus or H minus R minus, then these plaques are coming from recombinant phage. Right. So if you want to calculate the recombination frequency between H and R genetic markers, you have to calculate the recombination frequency, which would be H plus R plus plaque number plus H minus R minus plaque number divided by total plaques. So the total varieties divided by the recombinant varieties divided by the total varieties. I mean the number corresponding to each of those varieties. So this is basically how in Harsh's cross they try to calculate or they try to determine recombination. In this particular snapshot, you can see that there are different types of plaques. So this is a typical H plus R plus plaque, small and hazy. This is R minus but H plus, so large and hazy. This is R plus H minus, so small but clear. And this is R minus H minus, large and clear. So these are the different types of plaque that can be observed by the group, and based on counting these plaques, they were able to calculate the recombination frequency. So what was the result? They found out that actually this is their experiment. So this is R minus H plus, this is H minus R plus, 2 phage. Simultaneously, they are infecting this E. coli, and then they are crossing, and presumably they are producing H plus R minus. It is a parental one. H plus R plus, which is recombinant one, H minus R minus, again a recombinant one, and H minus R plus up second parental one. So if these 4 types of phages can be produced by this co-infection, and they actually calculated the number of phages from the plate that they experimentally obtained, and they found out that according to the plaque numbers, H minus R plus, H plus R minus, H plus R plus, and H minus R minus, the numbers were 42, 34, 12, and 12 respectively. Okay, and this means that from the plaques, 76% were of parental progeny, but 24% are of recombinant progeny. So the recombination frequency is 24%, which means that H plus and H and R, these two loci, if indeed they are determining these two traits, they are 0.24 map units distant. So these are 0.24 map unit distant because the recombination frequency between them is 24%. So through this model experiment, this setup, it was observed that not only we can actually visualize recombinant phage, but also we can calculate the recombination frequency. Now, what is the advantage of this kind of setup? First of all, phage infection and bacterial growth, these kind of things, they take much smaller time. Secondly, bacteria and phage, this infection system is much less complex than the huge large genomic content of Drosophila or any other bigger models. So these are highly reproducible. Reproducibility means if you repeat the same experiment, the deviation between the observations are less. So good experimental models always generate data which is reproducible. So that means because these are less complicated ones, so this is a very elegant model for calculating recombination between two loci, particularly for the phage biologist, because at that time phage biologists were trying to identify different genes. They were trying to build up genetic maps for different phages. So for that purpose, this kind of concept and this kind of observation became one of the highlights of phage research at that time. Another group, actually, it's a very important group, Seymour Benzer's group, because his contribution in genetics is in microbial genetics particularly is huge, and he is a scientist who is hailed as an individual who enabled the transition of genetics to molecular age, uh, who actually who actually uh, uh, contributed in such an extent that actually that enabled the transition of genetics to molecular age. It is, I'm quoting it, the quote that is provided here is from one of a write-up on him, and there it is written that more than any other single individual, he enabled the geneticists to adapt the molecular age. So that means the transition of genetics to molecular biology, one of the major contributors was Seymour Benzer. And huge contributions he made. And one of the major contribution is experiments for identifying different genetic markers for mapping phage genome loci. So the basic principle of the kind of experiments that he did is very similar to what we have already discussed. It is that if you are crossing two phage mutants for different characteristics, say the characteristics are minus plus, and the mutants are M1 and M2. So if these mutations are distantly located, then the chance of recombination would be high. If these mutations are closely related, then the chance of recombination will be less. Another principle is it's shown here. If here there is a mutation, here there is a mutation, the chance of recombination is less because the amount of DNA lying in between is less. But here, because the amount of DNA lying in between is high, the chance of recombination is high. And if you look into this diagram, say M1 and M2, these are two mutants, where in this particular position it's minus, here it's plus. So, and here it is plus, it's minus. So these two can be recombined, and after recombination, they can produce a DNA with both plus and produce another DNA with both minus. But if M1 and M2 are two mutants where in this particular loci or very closely located loci there is a mutation, that would mean that for this particular gene it would be minus minus both the mutants, and here there is no mutation in both the gene in both the mutants, so it's plus plus. So after recombination, because this minus mutation is so closely linked that there can be no cross in between, almost virtually there can be no cross in between. So the product will always be this minus plus and minus plus. So there will be no recombinant. So in principle, if two mutations are too closely related to one another, there will be no recombinant. If they are sufficiently distant to one another, there will be recombinant. So a recombination of frequency can help us to map different mutations on a genome. So what Benzer did, Benzer's test is one of the most important tests conceptually as well as in terms of its significance in understanding the molecular nature of phage genome or any genome per se. So Benzer's test, actually the system or the main target was the R phenotype of phage, what we have already discussed. So in their laboratory there was a collection of huge number of different mutants of T4 phage. I mean of different phage of course, but T4 is the model for this experiment, so we would concentrate on T4 phage. And they identified two sets of mutants which they named as R1 and R2 mutants. They found out that R2 mutants are better to study as there is a signature characteristics of these R2 mutants, and therefore they thought, okay, let us try to map the R2 loci in the phage genome through this mutant based and recombination based mapping, what we have already discussed. Now they found out that these R2 mutants, they are kind of conditional mutants. Conditional mutant means that the phenotype of the mutation can be observed under certain condition, and the phenotype is basically absent in some another condition. So although the mutation is there in the gene, it is not reflected in the phenotype unless a certain condition is provided. So for this R2 mutants, they identified that two E. coli strains that can act as host for T4 phage, but for these R2 mutants, their infectivity to these two hosts are different. So for T4 wild type, they can infect both E. coli B strain as well as E. coli K strain. So E. coli B strain can be infected by both mutant and wild type, but E. coli K strain can only be infected by wild type. The R2 mutant cannot infect E. coli K. So E. coli K is a kind of restrictive or non-permissive condition for R2 mutants, while E. coli B is a permissive condition for R2 mutant to grow. Okay, so based on these typical signature characteristics, they thought that, okay, let us try to map these R2 loci in the genome of phage, because in my last slide, I discussed that we can map different mutants. So if we have a collection of mutants corresponding to this R2 phenotype, so where is how distant those mutations are? That was their attempt to, that was their basically intention. So let us see how they did it. So they took different mutants, like here you can see one mutant is R1 and another mutant is R2, something like that. So here you see in the first phage, there is one mutation R1 which is mapped here. And in the second phase, there is one mutation here. So if you cross between these two, and if you see that they can replicate and they can recombine, then in the progeny, what would be the outcome? The outcome would be either parental R1, and this is a second loci which is plus for both. So here the mutation is in these genes, so R1. So R1 and plus wild type, R2 and plus wild type, again parental, and R1 and R2, this can be the third one. This is the double mutant. And then this R1 and R2 cross, which is leading to plus for both. So it is becoming some plus plus. So completely wild type. So two parentals and two recombinants that can be generated by this particular cross. Now, what they did? They infected E. coli B as well as E. coli K strain with these two fudge. Now in E. coli B strain, all of these mutants will be able to form plaque because E. coli B is permissive to all. But in E. coli K, only the wild type will be able to form plaque. The other varieties, R1 plus or R2 plus or R1, R2, both mutant, they will never be able to produce plaque. So, what would be the outcome? The outcome will be the strain B will give you count of all possible, total count of fudge, and this plate, it will give you count of the wild type. So now because the wild type is only half of the recombinant population, if we consider recombination frequency equivalent for all, so if it is half of the population, then the number of wild type would mean that this is the half number of the total number of recombinants. So the number that is obtained on this plate with E. coli K multiplied by 2 divided by the total number of plaques on plate B would be the recombination frequency. That was their basic concept. So what can be done, or what actually, sorry, what they found out is that using this kind of a recombination experiment, say multiple. Okay, so after standardizing this particular system, what they did, they attempted to map the R2 loci with their collection of mutants. Now, as you can see, this was the map that they prepared. Each of these blocks represent a specific independent mutation. And you can see there are spots like one here, where a bunch of independent mutations were mapped by them, and they assigned those loci as hotspots of mutation. Now, they performed some calculation to identify the minimum length or minimum distance that is required for recombination. And they found out that as per their experiment, as we discussed in the previous slide, that it is 0.1% was the number of wild type that they could detect. So in reality, the number of recombinant, the frequency or the rate of recombinants would be 0.2%. So that would mean that between two R2 mutants, there is 0.2% of distance, 0.2% of what? 0.2% of the map units. Now, according to their calculation, the total map unit for T4 was 1500. And we know a T4 genome is 2 into 10 to the power 5 base pairs long. So a simple calculation comes here. If 1500 map units corresponds to 2 into 10 to the power 5 base pairs, one map unit would correspond to 2 by 15 into 10 to the power 3 base pairs, and zero point 0.2 map unit would correspond to 0.02 map unit would correspond to 0.02 into 2 by 15 into 10 to the power 3 base pairs. That means 40 by 15 base pairs, which is equivalent to 3 nucleotides. So with their experimental setup, they could identify that the minimum distance for recombination would be 3 nucleotides. Later on, further experiments by Janowski and other, it was shown that recombination can occur in between any DNA. So actually, recombination can occur within a distance of 1 nucleotide too. So that was determined later on. But imagine the elegance of the experiment, that with this kind of setup, with least technological assistance like DNA sequencing or something, they were able to map the minimum distance required for recombination as low as 3 nucleotide, which is quite closer to the actual distance, which is 1 nucleotide. So this is an excellent system, excellent experiment through which actually they were able to show the event of recombination and its relation with the distance between the nucleotides. In a similar way, they also prepared a mapping system. They designed a mapping system that is called deletion mapping. The basic concept is, say, if you have a mutation in one of your T4 loci, R2 loci, and if you have a collection of deletion mutants, so if this deletion overlaps with this mutation, then you will not be able to get the wild type by recombination. But if they are distant, you will be able to have the wild type by recombination. Similarly, if you have two deletion mutations which are overlapping with one another, then with recombination you will never be able to have the wild type. But if the deletions are distant, then you will be able to have some recombination in between, and therefore you will be able to obtain some wild type recombinants. So using this particular principle, they designed a deletion mapping system. Just to make you understand the deletion mapping system, say if you have strains like 1, 2, 3, 4, 5, 6, each have some deletion in the genome. And now, if you have to uh, cross these strains, that means you have to co-infect the bacteria with these strains. So if you co-infect the bacteria with the same strain 1 and 1, then of course there will be no recombinant. Then if you co-infect with the bacteria with 1 and 2, then only in one case you will be able to have some recombinant if they do not overlap. So if you co-infect 2, 3, 4, 5, 6 with 1, and if you get only recombinant in 3, that would mean that only 1 and 3 are the deletions that do not overlap. Similarly, for 2, if you co-infect with 1, 3, 4, 5, and 6, and if you get recombinant for 3 and 4, that would mean that 4 is away from 2, and 3 is also away from 2. Okay. Similarly, for 3, if you can get recombinant only with 4 and 5, that would mean that 1, 2, and 6, they are overlapping with 6, overlapping with 3, and 4 and 5 are non-overlapping with 3. Similarly, for 4, if you get colonies plugs only in 5 and 6, that would mean that 4 is distant from 5 and 6, but it is overlapping with 1, 2, and 3. If you are not getting any colony in 1, 2, and 3 when it's co-infected with 4. Similarly, for 5, if you are not getting any colony with any other, say 1, 2, 4, 5, 3, 4, 5, no colonies are there. But if you are sorry, for 1, 2, 4, 5, you are not getting any colony, but you are getting colonies with 5 in case of 4. So 3 with 5, it is plus, so you are getting some colonies, and with 6, so 6 and 5, it's plus. So that would mean that 3 deletion is overlapping with 5 and no, so that would mean that deletion 3 is non-overlapping with 5 and 6. So with this kind of data, you can construct a deletion map like this. See, 1, it is overlapping with 2. So 1 and 2, there is no colony. Then 4 is overlapping with 1. So 1 and 4, there is no colony. You can see 0. And 1 is overlapping with 5. So 1 and 5, there is no colony. And 1 is overlapping with 2, 4, and 5. So see, there is no colony for 1. Now 3 is non-overlapping with 1, 2, or 4. So 3 is giving you a plus for 1, plus for 2, and plus for 4, and plus for 5 as well. So 3 is non-overlapping with all of these. But 3 is not giving any recombinant with 6, so that would mean 6 is overlapping with 3. Now for 6, you were not getting any colony for 1, any colony for 2, 3, but you were getting colony for 4 and 5. So that would mean 6 is overlapping with 1 and 2. In this segment, they are overlapping, but with 4 and 5, they are distant. So cumulatively, we can build up a map which looks like that. Here, 4 will be in one termini and 3 will be in one termini, and 1, 2, 5, and 6 will be arranged like that. So in this process, you can build up a deletion map. So this deletion map preparation is this was again some elegant way to identify different deletion mutants, and whether the deletions are located on the same region of the chromosome or not. Those were identified by this phenomenon. But it is again dependent on the crossover or crossover between two different genomes. After identifying different types of recombinants, they performed a test that is called complementation test, and this is also called Benzer's cis-trans complementation test. What is cis-trans? Imagine these pink and blue as two independent mutant phage. If the mutant is located on the same gene or in the same loci, then we would call them as cis. See here, the mutations are located on the same loci. So these are cis mutants. But if the mutants are distant enough so that they are located in two distinct loci or two distinct genes, we would call them as trans. Now, what is the implication? If you have two genomes where the two mutations are located in two independent genes, and if you are co-infecting those with bacteria, then deficiency of this particular mutant for the R2A gene and deficiency of this mutant for the R2B gene will be complemented by vice versa. So R2A's deficiency will be complemented by this phage, and R2B's deficiency here will be complemented by the pink one. So the gene products will be there. This will produce a good copy of R2B. This will produce a good copy of R2A. So now these R2B and R2A both will be available for both the phages, and therefore they will be able to produce plugs if the mutation is in cis position. But if the mutation is trans conformation, then what transposition, then what would happen? This phage and this phage both has mutation, They can be in different position of the same gene, but both are producing a non-functional version of R2A. So neither of these will be able to produce a good copy of R2A, which is required for the bacteria to either replicate or maybe to lyse, for the phage to replicate or maybe to lyse the bacteria to form certain number of plaque. So this clearly indicates that if the mutations are located on the same gene, that is if they are cis, there will be no plaque. And if the mutation is located in two different regions, that is if they are trans, there will be plaque. So with this, you'll be able to identify how many genes are there. If you have a collection of library and if you are performing this experiment here, you'll be able to identify how many genes are there. If mutant 1 and mutant 2 are complementing, that would mean two mutations are located in different genes. If the mutations are on the same gene, they will not complement. So conceptually, these will be able to determine the number of mutants. That was basically number of different genes from the collection of mutants. So this was the objective of complementation test. Now remember, complementation is not really recombination. Here, the DNAs are not crossing over with each other. The DNAs are independently expressed. Both the genomes are independently expressed. So there is no need of crossover or something. But phenotypically, it looks like recombination. But complementation actually is not recombination. It is a non-recombinational process where the phenotypic features are dependent on production of the gene products, and complementation is complementation of the deficiency of a gene product by another mutant. So the cis-trans complementation test is also something that was very important, but that is not mediated by recombination. So with all of these experiments, Benzer and his team were able to coin three different terms. One was recon, that is the unit of recombination. One was muton, which is unit of mutation. And they also defined cistron, which is unit of cis-trans complementation. Now, for cis-trans complementation, as I mentioned, the unit is basically a gene where loss of function mutations for if they are located on the same gene, it cannot complement. If they are located in two different genes, they can complement. So, cistron is nothing but a gene. Muton is basically one base where a mutation can occur. So it's basically a single base. And recon is also a single base because recombination can occur within a distance of a single base. Although at that time when they performed the experiment, they identified three nucleotides as the minimum distance required for recombination. Later on, we corrected it as recombination can occur between even a distance of one base pair. So this is how the classical experiments by Benzer and others they provided us novel insights into recombination, not only recombination, mutation, and processes like complementation. So with this set of discussion, I would conclude the primary experimental part of recombination. Next segment, I will discuss about mechanism of recombination, and I will discuss about some state-of-the-art findings on recombination as well as some important experiments associated with deciphering the mechanistic aspect of recombination.